So it is with my great enthusiasm that I introduce our next speaker, Dr. Laura Hack, clinical instructor and founding director of the Stanford Translational Precision Mental Health Clinic. Her focus is on identifying targeted treatments for subgroups of depressed patients. Here today to present on precision psychiatry and novel therapeutics informed by neuroscience, I welcome Dr. Hack.
Thank you so much, Hasna, for the kind introduction, and thank you all for coming. It's really an honor to be presenting as part of this distinguished panel. I'm thrilled to be here today to discuss the work we've been doing in the center on the clinical application of neuroscience-informed precision psychiatry. So while the implementation is more complicated, the goal of precision psychiatry is simple. It is to relieve patient suffering by matching preventative strategies and treatments to individual characteristics. Some have expressed concern, and rightfully so, that this high-tech approach will replace our current approach and take the human touch out of psychiatry. At the center, we don't see it as replacing our current approach to patient care, but instead complementing and improving upon it. And to illustrate this, I'll discuss precision psychiatry in the context of our current practice for depression, which is the focus in our center in 3 phases: data gathering, treatment decisions, and monitoring and prevention. So when a patient first presents with depressive symptoms, we currently already gather clinical information from multiple sources, including interview, observation, and sometimes questionnaires. We assess environmental exposures, and we may conduct limited biological measurements, such as thyroid hormone, to rule out medical causes. Precision psychiatry adds to that multiple other sources of data that we can now measure due to technological advances, including pharmacogenomics, inflammatory markers, and functional MRI, which you've heard about from Dr. Williams and Dr. Totsi. So in standard care, we then select among the first-line evidence-based antidepressants based on a limited number of factors, such as clinical experience or patient preference. With precision psychiatry, the vision is to match treatments with individualized profiles. So with this approach, we may start with therapies that are traditionally only considered later, such as TMS or transcranial magnetic stimulation or ketamine. Finally, in terms of prevention and monitoring, in the current practice, we typically follow up with patients every 1 to 3 months. With new technologies, we will now be able to monitor patients with real-time data collection, such as some of the sensors that Dr. Williams described. And this will allow us to intervene sooner, potentially before someone relapses. So at the center, we truly believe it's the right time to start applying precision psychiatry to the clinic. And we feel fortunate that the National Institute of Mental Health and Veterans Affairs agrees. So the NIMH develops a strategic plan every 5 years. And one of the objectives of the 2020 plan is tailoring existing interventions to optimize outcomes. Precision psychiatry is also a priority for the VA. In the fall of last year, Congress passed a bill called the Precision Medicine Initiative with the aim of identifying and validating brain and mental health biomarkers. At the center, we also believe that Stanford is the ideal place to be implementing precision psychiatry in the clinic. This is because precision health has been a focus at Stanford for many years, with Dean Miner leading the charge, as exemplified in his eloquent book, which Dr. Williams has mentioned. This text, High Tech and High Touch, is pulled directly from the Precision Health website at Stanford, and emphasizes the key point that while precision health and psychiatry involve advanced technologies, at Stanford and in the center, we are making every effort for the human aspect to remain. So in focusing on depression specifically, We know that a major issue is that it is highly heterogeneous, symptomatically and biologically. But for the most part, our first-line medication treatments have the same mechanism of action. As I've mentioned, we have some ways of tailoring treatment, but it is mostly a trial and error process, selecting among first treatments, then among novel and alternative treatments. This results in only about 30% of patients remitting on the first treatment and 60% after subsequent trials. Treatment resistance is associated with a significant increase in suicide, which we know is a major public health crisis, as exemplified by this WHO statistic that one person dies every 40 seconds by suicide. In the time that it will take for me to do this presentation, 30 people will have died by suicide. We really need to find better ways to do better for our patients. That's why we're thrilled to be launching the Stanford Translational Precision Mental Health Clinic, which Dr. Roberts announced. We sincerely thank her for making this clinic possible at Stanford. To our knowledge, our clinic is the first consultation clinic for patients with treatment-resistant depression, utilizing functional MRI as part of the evaluation and treatment planning process. Because this is so new, we will first launch the clinic as a research clinic under an IRB-approved protocol. The process will involve a comprehensive psychiatric evaluation, and testing may include standard blood work, inflammatory markers, pharmacogenomics, neurocognitive testing, and structural and functional MRI. We'll then prepare a feedback report with the data from all forms of testing. You'll hear from Dr. Wintermark in the upcoming presentation about the steps that will go into the preparation of this report. We will then review feedback with patients and suggest possible treatment options. If eligible, patients will also have the opportunity to enroll in our biomarker-guided trials, which I'll mention in a moment. So in the center, we've been focusing on testing targeted treatments for two subtypes of depression that we refer to as anhedonic and cognitive dysfunction subtypes, because they don't respond well to conventional antidepressants. And they are associated with an even higher risk of suicide and poor psychosocial functioning than other types of depression. So I'll start with the anhedonic subtype of depression. To give you a sense of anhedonia, if you have not experienced it before, which I hope you have not, please think for a moment about things that you love doing or that bring you joy. For example, spending a day at a warm beach with a refreshing breeze, your child's beautiful smile, or some delicious chocolate. Now imagine not having any enjoyment of these activities and the lack of motivation and guilt that may go along with that. This is what individuals with anhedonia experience. Most simply, anhedonia is a lack of pleasure, but it may also involve a lack of interest and motivation for enjoyable activities. It occurs at a clinically significant level in 37% of patients with depression. There's evidence that anhedonia is associated with low activity of the ventral striatum, which is a key node in the positive affect circuit, which Dr. Williams discussed. So this region receives input from dopamine neurons. The question becomes, how might we target dopamine receptors in the ventral striatum? So it turns out that there's a certain type of dopamine receptors called D3 receptors that are densely localized in the striatum. There's also an FDA-approved drug for Parkinson's disease and restless leg syndrome called Pramipexole, or Mirapex, that preferentially binds to these D3 receptors. There's evidence that Pramipexole increases activity in the ventral striatum in healthy subjects. So we conducted an 8-week pilot study of Pramipexole in participants with depression and high anhedonia. I wanted to share with you the case of Mo, a participant in our pilot study. His story was featured in Science magazine, and you can see him pictured here with myself and Dr. Williams. So Mo enrolled in our study at the age of 37, at a time when he was in a deep depression with a high level of anhedonia. He had lost his job, he was no longer engaging in activities that he previously enjoyed, and he began to isolate himself. After 8 weeks of treatment with Pramipexole, we found that there was an increase in brain activity in that key reward region, the ventral striatum. Along with this, his depression level improved from moderately severe to remission, and there was an early decrease in symptoms within 2 weeks. Also, his anhedonia decreased by over 30% from the maximum level. His suicidal thoughts resolved and his sleep improved. He had a very eloquent way of summarizing the change. He said, "The color has come back into my life. I enjoy music and food again." So now turning our attention to the other subtype of depression that we've been focusing on. As many as 90% of patients report cognitive impairments, and 38% show deficits on objective testing. In the large international multi-site study of biomarkers in depression that you heard about from Dr. Williams, poor performance on a cognitive task was associated with low activity in a key region of the cognitive control circuit, which Dr. Williams and Dr. Tosi have beautifully described. This region is the dorsolateral prefrontal cortex, shown here with the circles. It's important for executive functions such as planning and working memory. So how might we tailor treatment to a cognitive subtype of depression characterized by low activity in the dorsolateral prefrontal cortex? Well, we know that there's a high concentration of something called alpha-2 receptors in the DLPFC. And there's an FDA-approved drug called Guanfacine, which selectively binds to alpha-2 receptors and strengthens the connections in the prefrontal cortex. Guanfacine is FDA-approved for ADHD and hypertension under the brand names Intuniv and Tenex. It has been shown to improve performance on a working memory task and increase activity in the dorsolateral prefrontal cortex in patients with mild traumatic brain injury. In collaboration with Dr. Banstra, a psychiatrist in the department, we ran a pilot study of Guanfacine in depression. I'd like to share with you one of the cases. Mrs. A is a 52-year-old female who was currently in a major depressive episode, despite currently taking an antidepressant and an augmentation agent. She reported to Dr. Banster problems with motivation and concentration. She said, "I haven't been able to concentrate on work for a month now. I haven't put out anything productive. I find myself easily distracted." Dr. Banster prescribed 1 milligram of Guanfacine daily for a week to get her used to the drug, and then increased this to 2 milligrams daily. Over the course of the 8-week study, Mrs. A's depression decreased from a moderate level to a level that is considered in remission. On objective testing, assessing a measure of planning, that Mrs. A revealed that Mrs. A had a substantial improvement in this measure. Most importantly, Miss A had the motivation to return to work during the pandemic, even when she didn't have to, and was able to focus on projects again. She also had the motivation to start planning a vacation. She continues to take Guanfacine 6 weeks out and continues to do well. So we're thrilled to be launching what we call the biomarker-guided study of depression later this year. What we plan to do is recruit subjects with anhedonia or prominent executive function deficits that either have high or low activity in the ventral striatum or the prefrontal dorsolateral prefrontal cortex. We'll then randomize each group to Pramipexole or Guanfacine or placebo for 8 weeks of treatment. We hypothesize that we'll see more change in the ventral striatum or prefrontal cortex activity in the low baseline group, as well as greater improvement in cognitive performance, symptoms, and quality of life. So during this presentation, I've discussed Pramipexole and Guanfacine for 2 subtypes of depression. In the center, we are also looking at other novel and experimental therapies for specific subtypes of depression. For example, I'm a clinician in the Precision Neuromodulation Clinic at the Palo Alto VA, and we have a study examining the effect of transcranial magnetic stimulation, or TMS, on cognition in depression. In the clinic, I also helped recently launch intranasal esketamine as another treatment option for veterans with treatment-resistant depression. This therapy may be particularly helpful for depressed patients with dysfunction in the default mode network. While there is evidence that the psychedelic MDMA may be helpful for dysfunction in the negative affect network, which you've heard about from Dr. Williams. In the center, we are running studies of ketamine and soon MDMA in healthy subjects to help us understand more about how they alter brain neurocircuitry in an effort to eventually be able to tailor these treatments. So I'm delighted to let you know that Dr. Williams and I have co-edited the first textbook on precision psychiatry, and this will be coming out next month. We are fortunate to have had multiple leaders in the field contribute to this book. This book highlights the current and potential applications of multiple tools, including cognitive testing, inflammatory markers, and of course, neuroimaging into the clinic. With that, I'd like to thank you so much for your attention. I'd also like to express my sincere appreciation to the participants, our funding sources, my mentors, several of whom are here today, including Dr. Williams, O'Hara, and Wintermark, and our wonderful lab members who are pictured here with their favorite animals, and of course, to our incredible director and the person who made this center possible, Dr. Williams. As others have highlighted, we very much welcome your participation and partnership. Thank you again for your attention.
Thank you, Dr. Hack, for sharing your exciting pilot data and plans for innovative biomarker-guided trials for subtypes of treatment-resistant depression. We'll now move to the Q&A portion of our program, and if you have any questions, please add them in the chat. And we actually have one coming in for you. Would focusing too much on the brain be a disadvantage to depression research?
Yeah, thank you for that question. So I just wanted to emphasize again how we're really taking a holistic approach to this and incorporating a number of different measures into our consideration of potential treatment options for patients. And so, you know, I definitely do understand a concern about too much focus on biology, but I want to emphasize that in the assessments that we're going to do of patients in the clinic, it's going to involve definitely talking to the patients and you know, all of the standard things that we do in psychiatry in addition to the brain. So I really think that that combination is going to help move us forward in psychiatry. And just one other study to mention was one that was actually conducted at Stanford, where researchers found that patients really would be interested in hearing about their brain measures, and this They felt like this would lower stigma, help them understand their condition better, and then, of course, ideally help guide treatment decisions.
Thank you, Dr. Hack. We have time for one final question, and then we can ask some others in the panel discussion. Thanks for your excellent presentation. What is the data for using Wellbutrin for anhedonic depression?
Yeah, that's a great question. So there is data that Wellbutrin is helpful, but the issue with it is that it's not targeted or specific. And so we hypothesize that Pramipexole may even be more helpful for a subgroup of people who really have both the anhedonia as well as dysfunction in the ventral striatum.
Thank you, Dr. Hack. Thank you again for your presentation and answering our questions. We'll ask some more in the panel discussion.